Hi, my name is Cameron Dyer, and I'm going to explain what developmental networks are in developmental genetics and give an example of a developmental network using fictional characters. First, a definition. A 2005 article by Michael Levine and Eric H. Davidson in PNAS states that developmental networks are logic maps that state in detail the inputs into each cluster of DNA sequence elements that serve as target sites for transcription factors, so that one can see how a given gene is fired off at a given time and place. They also provide specifically testable sets of predictions of just what target sites are hardwired into the DNA sequence. But what does this mean? This is a process by which an organism regulates processes related to its function and development, such as turning on or off genes that are only needed at one particular life stage or in a particular environment. The transcription of these genes are regulated by proteins expressed by other genes in the genome, which go on to bind to the genetic material as transcription factors. This process gives organisms a level of control over gene transcription and the ability to do things like differentiate different cell types or allow cells to be better equipped to survive in different conditions. To illustrate how this works, let's imagine a network consisting of 4 genes in an organism called the mushroom kingdom. We'll call the genes Mario, Yoshi, Peach, and Bowser. All 4 genes are located in different parts of the genome, and the transcription and translation of each gene in the sequence will create a protein that will either inhibit or stimulate transcription of another gene in the sequence. The developmental path is as follows. This means that the Mario gene can't be expressed until the Yoshi gene product stimulates it. In turn, the Mario gene product, once expressed, will go on to inhibit the Bowser gene. If the Bowser gene were to be expressed, it would inhibit the expression of the Peach gene. The expression of the Peach gene would create a product beneficial for the mushroom kingdom. In this example, we can imagine that this pathway might allow the mushroom kingdom to quickly address its needs at a specific point in time and in that particular environment. Maybe the product Peach is introducing is a new piece of legislation or giving an inspirational speech. As in all discussions involving genetics, mutations can occur and have drastic impacts on the phenotype of the organism. If this organism exhibits a mutation in the Bowser gene, that gene won't be expressed. Therefore, there will be nothing to inhibit expression of the Peach gene, so the Peach gene will be expressed and the mushroom kingdom will have peace and prosperity. If there's a mutation in the Yoshi gene, that gene will not be properly expressed and will not be able to stimulate expression of the Mario gene. This will result in the Bowser gene not being inhibited. Therefore, Bowser will be expressed and Princess Peach will be inhibited. Double mutations could also occur. Imagine if there's a mutation in both the Yoshi and Mario genes. The mutation in the Yoshi gene will not allow Yoshi to stimulate Mario, therefore Mario will not be able to inhibit Bowser, even if Mario didn't also have a mutation. Or another example, if a mutation occurs in both the Mario and Bowser genes, the Mario product will not be able to inhibit the Bowser gene, but the Bowser gene product will not be able to inhibit. Therefore, Princess Peach will be able to be expressed. The ability to create different proteins or not in response to environmental changes at any given time gives organisms incredible flexibility and adaptability and are vital to the proper development of organisms.